So, good day everyone. We're reviewing Wetzel's Limnology textbook. This is edition number 4. We are on chapter 14. And today we'll be looking at nitrogen sources, transformations, and fate in lakes and reservoirs. And let's get started. So, let's see. Nitrogen occurs in fresh waters in numerous forms. So, ammonium, nitrite, nitrate, dissolved molecular nitrogen, organic compounds ranging from amino acids and amines to proteins, and recalcitrant humic compounds of low nitrogen content. Sources of nitrogen include precipitation falling directly onto the lake surface, nitrogen fixation both in the water and the sediment, and inputs from surface and groundwater drainage, and losses of nitrogen occur via effluent outflow from the basin, reduction of nitrate to nitrogen by bacterial denitrification, and anamox, with subsequent return of nitrogen gas to the atmosphere, and permanent sedimentation loss of inorganic and organic nitrogen containing compounds to the sediments. Key processes and inputs outlined below are atmospheric inputs to lakes and reservoirs, including nitrogen fixation, nitrogen transformation processes within the systems, and atmospheric removal processes. Hydrologic outputs and inputs of nitrogen are also important, and are briefly outlined in the section on nitrogen budgets in lakes and reservoirs. Atmospheric inputs. The amount of influent nitrogen to lakes and their drainage areas from atmospheric sources is often considered to be minor in comparison with that from direct terrestrial runoff. However, nitrogen from precipitation and direct bulk particulate fallout can be extremely variable depending on local meteorological conditions, wind patterns, and the location of streams and lakes with respect to industrial and agricultural outputs. Globally, atmospheric nitrogen deposition has increased more than tenfold over the past few decades. It is estimated that in 1860, 34 teragram of nitrogen per year of nitrogen was emitted as oxides of nitrogen and ammonia, and then deposited to the Earth's surface in 1995. It had increased to 100 teragram of nitrogen per year. By 2050, it is projected to be 200 teragram of nitrogen per year. Now, China has become the largest global emitter of reactive nitrogen in the last 2 decades. The combination of higher atmospheric loads and the building of more reservoirs has doubled nitrogen inputs to lakes, rivers, and reservoirs in China from the 1990s to the 2010s. In Lake Taiyu, China, atmospheric deposition accounts for 12% of riverine nitrogen inputs to the lake. Nitrogen deposition into lakes, reservoirs, and rivers is having significant effects on nitrogen concentration and productivity. Nitrogen mass balance techniques applied to a large number of drainage basins and lakes in central Ontario, for example, indicated that on a regional area weighted basis, 67% of bulk atmospheric total nitrogen was stored or denitrified terrestrially, 12% was denitrified in lakes, 4% was stored in lake sediments, and 17% was exported from the lakes. For example, in relatively oligotrophic mountainous regions of granitic bedrock, precipitation is a major source of nitrogen. Nitrogen deposition in mountain lakes can also increase the rate of denitrification. Inorganic nitrogen input by precipitation similarly was found to be a major source of loading to the drainage basin of Lake Tahoe. A global analysis has shown that increased atmospheric nitrogen deposition in lakes as a result of anthropogenic activities may have shifted some lakes from nitrogen to phosphorus limitation. This impacts nitrogen cycling and higher trophic levels. Critical thresholds in atmospheric nitrogen for nutrient enrichment and acidification of lakes across the United States have been identified to guide water quality management. Atmospheric nitrogen may enter a drainage basin in many forms as dissolved nitrogen, nitric acid, ammonium, nitrite, as well as ammonium plus. So actually we have that twice. Also, ammonium adsorbed to inorganic particulate matter and as organic compound which can occur in either dissolved or particulate phases. No direct relationship exists between the volume of rainfall or snowfall and the quantity of nitrogen influx per area of land or water. Dry fallout can contain as much as 10 times the quantity of nutrients commonly found in rain. The nitrogen content of snow is often much higher than that of rain and can contribute up to half of the total annual nitrogen influx to a stream or lake, even though snow generally constitutes a small proportion of total precipitation. In non-polluted areas, most of the combined nitrogen in the atmosphere is ammonia, much of which originates from the decomposition of terrestrial organic matter. Atmospheric ammonia, associated with dust particles, can be oxidized to nitrate, so that precipitation contains both ammonium and nitrate. Nitrogen fixation. Nitrogen fixation, in which atmospheric nitrogen is utilized by microbes or cyanobacteria for growth, plays an important role in the productivity of streams and lakes, particularly where dissolved inorganic nitrogen is depleted. In the open waters of lakes, cyanobacteria that possesses heterocysts can be an important source of nitrogen. Heterocysts, also called heterocytes, are specialized cells that occur singly in some filamentous cyanobacteria and are the site of nitrogen fixation. Nitrogen fixation has also been found to occur in some unicellular forms that do not produce heterocysts. However, many species of cyanobacteria, such as Dolichospermum, previously Anabaima, numbers of heterocysts correspond approximately to the observed nitrogen fixing capacity. Nitrogen fixation is primarily light dependent in that the process requires reducing power and adenosine triphosphate, both of which are generated in photosynthesis. Electrons for the reduction of nitrogen are supplied by the dinitrogenase reductase in a highly endergonic reaction, a chemical reaction where energy is absorbed. That requires about 12 to 15 moles of ATP per mole of nitrogen reduced. Nitrogen-fixing photosynthetic bacteria can fix limited amount of nitrogen in the dark, but at rates usually less than 10% of maximum daytime rates. In full sunlight, nitrogen fixation is often inhibited at the surface. Below this, there is nearly exponential decrease as light attenuates. The pattern of nitrogen fixation rates through the water column is similar to that of photosynthesis. The use of nitrogen fixation for growth by cyanobacteria is energetically expensive compared to the use of dissolved inorganic sources such as ammonium or nitrate. Therefore, species will preferentially use the inorganic sources, and growth is typically faster when these sources are available. Additionally, alternating between nitrogen fixation and the use of dissolved inorganic sources may not happen immediately, particularly for species that lose their heterocysts when nitrogen fixation is not occurring. Nitrogen fixation has also been correlated positively with concentration of dissolved organic nitrogen occurring in the water. Algae and cyanobacteria release extracellularly many simple and complex organic carbon and nitrogen compounds. Heterotrophic bacteria colonize common nitrogen-fixing cyanobacteria such as Anabaima, particularly during the maxima of nitrogen-fixing blooms. These cyanobacterial, heterotrophic bacterial aggregates form internal microenvironments with macrozones of redox conditions distinct from those of the surrounding pelagic or benthic habitats. The oxygen-depleted macrozones adjacent to the heterocysts protect the highly oxygen-sensitive nitrogenous. In temperate winters, nitrogen fixation is non-existent or greatly reduced. However, in productive tropical lakes, where the periodicity of physiochemical factors and cyanobacteria is less marked, nitrogen fixation rates can extend throughout the year. Evaluation of nitrogen cycles of lakes or streams require estimates of the total nitrogen fixed per annum. Planktonic nitrogen fixation tends to be low in oligotrophic and mesotrophic lakes, generally way less than 0.1 grams of nitrogen per meter square per year, and may be higher in eutrophic lakes, 0.2 to 9.2 grams of nitrogen per meter square per year. Benthic fixation in oligotrophic ecosystem is often dominated by cyanobacteria and tends to increase in cyanobacterial mats of very productive shallow environments, 1 to 76 grams of nitrogen per meter square per year. Despite the relatively small area of such their nitrogen contribution to the entire ecosystem can be disproportionately large. Nitrogen fixation may be insufficient at balancing system nitrogen deficit in lakes and reservoirs, and therefore nitrogen will not accumulate relative to phosphorus. A study using a mass balance model suggested that approximately half of oligotrophic lakes in the United States had a stoichiometric nitrogen deficit relative to phosphorus, while a higher percentage of eutrophic and hypereutrophic lakes, between 72 to 89%, had a similar nitrogen deficit. The process of nitrogen fixation cannot be viewed in isolation. Phosphorus is a critical element needed to support nitrogen fixation rates. Rates of nitrogen fixation are greatly enhanced when the productivity of a lake is increased by phosphorus fertilization. Nitrogen fixation capabilities are widespread throughout other photosynthetic bacteria. Heterotrophic nitrogen fixation is commonly disregarded on the premise that these nitrogen-fixing bacteria are limited by the low availability of exogenous carbohydrates. About 1 to 25 milligrams of nitrogen can be fixed per gram of carbohydrate utilized. Sufficiently large quantities of soluble carbohydrates are rarely available in natural waters, and there is an intense competition for these substrates by heterotrophic bacteria incapable of nitrogen fixation. The most common heterotrophic nitrogen-fixing bacteria comprise several species of Azotobacter and Clostridium pastorianum, while several methane-oxidizing bacteria are also capable of nitrogen fixation. In addition to these inputs of nitrogen from drainage runoff from terrestrial sources, lakes and streams often are bordered by dense stands of shrub-like trees, some of which can fix nitrogen from the atmosphere. Common species of the genera Alnus and Myrica are non-leguminous angiosperms that form large nodules containing an actinomycetal fungal endophyte or at or just below the soil surface. Alnus trees have been implicated as a significant nitrogen source for streams and lakes in glaciated regions of Alaska that are particularly nitrogen deficient. A New Zealand study found that invasive nitrogen-fixing species such as European gorse can be a significant nitrate contributor to rivers and creeks compared with native forests, thus impacting water quality. Microbes and cyanobacteria attached to macrophytes can also be a significant contributor of nitrogen to waterways. A study of the wetland sedge, Chenoplectus californicus, found that microbes associated with the plant contributed fixed nitrogen to increase the nitrogen content by 13.8 to 32.5%. The aquatic fern Azolla contains the symbiotic heterosis-forming nitrogen-fixing cyanobacteria Anabima azolae. Azolla species are actively cultivated in rice agriculture to supplement alternative sources of nitrogen fertilization, nitrate reduction, and denitrification. Microbes can reduce nitrate to nitrogen and N2O through denitrification and anaerobic ammonium oxidation, (ANAMOX) pathways, or to ammonium through the dissimilatory nitrate reduction to ammonium, DNRA pathway. Microbes involved in denitrification, ANAMOX, and DNRA therefore compete for the same substrate and can coexist in natural environments, including both sediments and the water column. The assimilation of nitrate and its reduction by aquatic plants can be a dominant process of nitrate removal in the trophogenic zones of lakes. As much as 60% of the photo-assimilated nitrate can be excreted as dissolved organic nitrogen compounds, some of which are simple amino acids readily utilized by microbes. However, nitrate and nitrite can also be denitrified by bacteria producing nitrogen gas with concomitant oxidation of organic matter. The general sequence of this process is nitrate to nitrite to N2O to dinitrogen, which results in a significant reduction of combined nitrogen that can in part be lost from the ecosystem. Many facultative anaerobic bacteria, particularly of the genera Pseudomonas, Acromobacter, Escherichia, and let's see here, and Bacillus and Micrococcus can utilize nitrate, the denitrification reaction is associated with the enzyme nitrogen reductase and requires the cofactors iron and molybdenum. Denitrification operates similarly under both aerobic and anaerobic conditions. So before we move further, let's look at this figure here, figure 14-4. So we have a conceptual diagram of nitrate removal pathways. Blue arrows denote autotrophic, which means that microbes use inorganic material for energy and nutrients. And then pathways, while purple arrows denote heterotrophic. Microbes use organic material for energy and nutrients. So we have pathways, we have this Fe2, which is ferrous, and we have Fe3+, which is ferric, and we have H2S, which is hydrogen sulfide, and SO4 2-, which is the sulfate. So let's see, we have, in the middle of it, we have the nitrate, and so we have a pathway for nitrite, and nitrite plus ammonium is equal to dinitrogen, so through the NMOX pathways. Then we have nitrate that gets reduced into nitrite, and then through iron-driven denitrification, so where Fe2+, which is ferrous, gets into a ferric iron, and so therefore loses 1 electron, and it gets reduced to create nitrogen gas or dinitrogen. Nitrate, then here we have the purple arrows. So the purple arrows are heterotrophic, so where the microbes use the organic material for energy rather than they use inorganic material for energy. And so in this case we have the fermentation pathway that results in ammonium, and then we have this organic carbon that releases carbon dioxide, and that results in dinitrogen and N2O, and which is the respiratory denitrification. And then of course we also have something, the black arrow, which is neither an autotrophic or an heterotrophic process, where we have the assimilation of nitrate through biomass and so sequestration. So an exemplary reaction of the oxidation of glucose and the concomitant reduction of nitrate. So we have glucose plus 12 nitrate is equal to 12 NO2 plus 6 carbon dioxide plus 6 waters, and a delta Gibbs energy of negative 1924.3 kilojoule per mole. And for the reduction of nitrite to molecular nitrogen, we have glucose plus 8 nitrite is equal to 4 nitrite plus 2 CO2 plus 4 carbonates plus 6 waters, and the Gibbs energy for that is even higher, which is negative 3012.5 kilojoule per mole. Approximately as much free energy results in the aerobic oxidation of glucose by dissolved oxygen of Gibbs energy of negative 2924.6 kilojoule per mole. The denitrification reaction occurs intensely in anaerobic environments, such as that in the apolimnia of eutrophic lakes, and in anoxic sediments where oxidizable organic substrates are relatively abundant. However, aerobic denitrifiers have also been found in natural environments, and some bacteria can simultaneously nitrify and denitrify under aerobic conditions in the water column. In eutrophic lakes, littoral areas, and wetlands, denitrification is a major process influencing the distribution of nitrate. A range of organic carbon compounds commonly found in agricultural and domestic pollutants provide the energy source in the nitrate reduction. Glucose acetates and particularly formate, among others, have been shown to induce high nitrite accumulation caused in part by partial inhibition of denitrification. Nitrate can also be denitrified concurrently with the oxidation of sulfur. The process is accomplished by denitrifying sulfur bacteria, particularly Thiobacillus denitrificans, that utilize sulfur or reduced sulfur compounds such as thiosulfate. So we have 5 sulfur here with, so that would be potassium nitrate plus 2 waters is equal to 3 dinitrogen plus potassium sulfate plus 4, like this is potassium hydrosulfate, I believe, plus 5 Na2S2O3. So this is a sodium sulfide plus 8 potassium nitrate plus 2 sodium bicarbonate. And then after that, that reaction then results in 6 sodium sulfate with 4 potassium sulfate, some CO2, water, and 4 dinitrogen. So ultimately what we are doing here, we are emitting nitrogen where we are starting with potassium nitrate. Both processes occur chemosynthetically under dark anaerobic conditions and yield relatively small changes in free energy. In a similar manner, the chemolithotrophic, which are organisms capable of using inorganic rather than organic molecules for fueling reactions, iron bacterium, Gallionella ferrugina, reduces nitrate to dinitrogen via the oxidation of ferrous ions under anaerobic conditions. So why don't we leave it here today, and tomorrow we pick it up with this reduction of nitrate, and we'll look at this at the contribution of iron in that pathway. So thanks for attending today's session on nitrogen sources, transformation, and fate in lakes and reservoirs, and I'll see you tomorrow, and take it easy. Bye.